Bismillahirrahmanirrahim. Assalamu alaikum dear viewers. I am Professor Dr. Muhammad Usman, Professor of Physiology, and today we are going to talk about a very interesting topic regarding the eye physiology, that is color vision. So basically we know that in our retina there are two photoreceptors, that is rods and cones. Now rods are rod shaped or cylindrical and the cones are conical in shape. Rods are meant for black and white or achromatic vision, and they are also important for the night vision, okay, while the cones, they are meant for chromatic or color vision, and they are also responsible for the bright daytime vision, okay. So according to the young Helmholtz theory of the color vision, there are three basic type of cones in our retina, that is red cones, green cones and blue cones. Red cones respond maximum to the light of wavelength 570 nanometers. Green cones respond maximum to the light of wavelength 535 nanometers, while blue cones respond maximum to the light having a wavelength of 445 nanometers, while the rods, they respond maximum to the light of wavelength 505 nanometers. Now the question arises that how do we see colored objects? Basically color vision depends upon the ratio of the stimulation of the three types of cones, okay. It depends upon the ratio of the stimulation of the three types of cones. For example, a person who is seeing a blue colored, now this blue colored object, it absorbs the red and green wavelengths of the light and it reflects the blue wavelength, and this blue colored light which it will be absorbed by the photopigment in the blue cones of the eye, thus activating them. Now white is a mixture of all the wavelengths of the light, whereas black is complete absence of the light. Now if our red cones are being stimulated 0% and green cones are stimulated 0% and blue cones are stimulated to the extent of 97%, then the color which we will see is blue. Similarly, if red cones are stimulated 83%, green cones are stimulated also 83% and blue cones are stimulated 0%, then the color which will be perceived by us will be, yes, it will be yellow. In the same way, if red cones are stimulated 31%, green cones are stimulated 67% and blue cones are stimulated 36%, then the resultant color will be green. It will be perceived as green by our visual cortex. So in this way the color vision, it depends upon the ratio of the stimulation of the three types of cones. Now let us talk about an abnormality related to the color vision, that is color weakness or color blindness. Now regarding this, we can divide the population into three types, that is trichromates, dichromates and monochromates. Trichromates are those individuals which have all the three cone systems, that is red, green and blue cones are present in these individuals, but they can be subdivided into two types, that is normal trichromates, these are the individuals with normal color vision, while anomalous trichromates have one weak cone system. They can have protanopia, deuteranomally or tritanopia. Protanopia is an anomaly in which the individual has weakness for red color and he has defective red cones. Similarly, deuteranomally is an anomaly in which the individual has weakness for the green color and there are defective green cones. Likewise, in tritanopia, the weakness is for blue color and the blue cones are defective. Then dichromates are those individuals which have two cone systems, while one cone system is absent. They are color blind person. They can have protanopia, deuteranopia or tritanopia. Now protanopes which have condition protanopia, they have blindness for the red color and red cones are absent. Deuteranopia is that form of color blindness in which the individual is blind for the green color and there are absent green cones. Likewise, in tritanopia, the individual is blind for blue color and he has absent blue cones. The person will be called tritanope. Now, the third type of individuals are monochromates and they have one cone system only, while two cones are absent in them. They have the ability to see only one color. Now, there are various causes for the color blindness and the most common cause of color blindness is genetic or inheritance. It is inherited in X-linked recessive form and there are defective options. The most common form of color weakness or color blindness is red-green weakness. It occurs almost exclusively in the males while female is said to be color blindness carrier. It has been estimated that 3% of all women they are color blindness carrier. Then another cause of the color weakness or color blindness is lesion of the visual cortex which is concerned with the color vision. A rare form of color blindness or color weakness is drug induced in which the drugs like sildenafil inhibit retinal as well as penile form of the phosphodiesterase enzyme, and you know that this is very important enzyme for phototransduction. So, it can result into transient blue green color weakness. Now, color blindness can be tested and there are various tests available for checking color blindness or color weakness, but the most common and easiest form of testing color blindness is ishihara charts in which different color plates are used. Likewise, you are seeing one color plate. Now, a normal individual he will read number 74 on this color plate, but a red green color blind person will read 21. Similarly, here in this chart a normal individual he will read 42. Okay, normal individual will read 42 while a red blind person protanope will read only 2 and a green blind person or deuteranope he will read only 4. Now, thanks for watching today's video. If you have liked this video, please like and subscribe to this channel. Happy learning to all of you. Stay blessed and Allah Hafiz.